Hi everyone, I'm Yun, a PhD student of the Australian National Insect Collection of CSIRO and the Australian National University. In this talk, I will talk about the taxonomic review of Miltotranes, the Bowenia-pollinated Cycad weevils, with discovery of a new species and new insights into Bowenia systematics. Cycads are tropical and subtropical pond-like gymnosperms. They diversified and are widely distributed during the Mesozoic. Some species of cycads are commonly used in horticulture, and they also have conservation values, as many species are listed in the Ocean Red List and threatened by habitat destruction and illegal poaching. Cycads are mainly pollinated by beetles, especially weevils. With nearly 90 species have been described, Australia is one of cycad diversity centers in the world. It belongs to the four genera: Cycas, Macrozomia, Lepidozomia, and Bowenia. And three of them are only endemic to Australia. Bowenia exhibits unique appearance with broad leaves. It has two extant species, Spectabilis in wet tropics in North Queensland and Cellulata in Byfield area of central eastern Queensland. Both of them are listed in CITES Appendix 2 and IUCN Red List at least concern. Miltotranes weevils belong to the Tranes group of Molytinae, exhibiting the small size length around 6.5 millimeters and the reddish color pattern. They show distinct sexual dimorphism. For instance, male has stronger femora and tibia, protibial brush, and large elevated erectile structure on prosternum. Miltotranes has two host-specific namespecies. One is Prostonalis pollinating Spectabilis, and another one is Subopacus pollinating Cellulata. Both species present the geographic distribution inconsistent with their host plants of Bowenia. In the field, Miltotranes adults congregate, mate, and lay eggs in the male cones during the annual coning period, and the larvae feed on its mesophyll tissue. The mature larvae probably leave the male cones after their disintegration and pupate in surrounding substrates. Miltotranes weevils may pass the period until the next coning season in the adult stage, and they may directly eclose in the end of coning period by the rainy season, without larval or pupal diapers, according to field observations and laboratory rearing in Shaw et al. 2020. There's still no phylogenetic work on Tranes group. However, our preliminary morphological characteristic analysis reveals that Miltotranes is close to the clade Tranes insignificant species group and Tranes latoroidal species group. Secondary colonization of cycad groups might occur within cycad pollinated Tranes group weevils, as Bowenia is unrelated with Macrozomia and Lepidozomia. In this study, we plan to review the generic characteristics and species identification based on the detailed morphological comparison, and generate a key to species based on our taxonomic results. The right side is our project diagram. Briefly, we combine the type specimen and the description to identify our material and summarize the taxonomy of Miltotranes. We examined the specimen from several museum collections and the type specimens of both named Miltotranes species from South Australia Museum. We combined the external morphology, terminalia, and the larval characters to do the species identification. The generic diagnosis is based on the comparison with other genera of Tranes group. The terminology for description follows Oberpreda et al. In 2014. So after our morphological comparison under a microscope, we summarized 28 adult characters from external appearance and terminalia and 7 larval characters. The combination of these characters could clearly separate Miltotranes from other Molytinae weevils. For species identification, all morphological study reveals that both named species are good species. They can be easily distinguished from each other by color pattern, male protibia, male elevated and setae structure on prosterna, left ventral of female, S abdominal sternet of male, and male genitalic structure. During our morphological examination, we noticed an undescribed species of Miltotranes from Macrores range of Cape York Peninsula. This species is similar to Postonalis, but can be easily distinguished by its male genitalia, including the shape of penis and the sclerize of endophallus. So, what we can learn from the discovery of a new species of Miltotranes weevils? This new species is associated with the local population of Bowenia cycad from Macrores range of Cape York Peninsula in far north Queensland, which is considered as Bowenia spectabilis for the time being, based on a morphological study in Wilson's master thesis in 2004. This putative spectabilis local population is separated from the true spectabilis in wet tropics by Norman Bay gap, which could be established from the late Miocene. Together with the host specificity of Miltotranes, it suggests this local population of Bowenia could be a new species, and further investigation is needed. Bowenia species Tinaru is another local population of Bowenia, distributed around the Tinaru area on the Atherton tablelands, showing the intermediate morphological characteristics between Spectabilis and Cellulata. Currently, it is considered as an ecotype of Spectabilis, supported by morphological characteristic analysis of Wilson in 2004 and the karyotype analysis of Coleoptera et al. In 2000 and 2001. Our taxonomic study on Miltotranes, its specific pollinators, reveals that both typical Spectabilis and the species in Tinaru share the same species of Weevil pollinator, providing another evidence for taxonomic identity of Bowenia species Tinaru from entomological side. So in this study, first of all, we confirmed 3 species in Miltotranes based on morphological comparison. And the discovery of new species suggests the diversity of Australian cycad-associated weevils is underestimated. Based on the biogeographic evidence and the pollinator host specificity, it suggests a potential new species of CITES-protected Bowenia cycads. Our taxonomic study on Bowenia-pollinated Miltotranes supports that Bowenia species Tinaru is a variation of Spectabilis as previously stated. It highlights new insights into cycad systematics from the entomological perspective. In the future, we wish to use genomic data to see if molecular species delimitation is consistent with morphospecies identification. And we also plan to do phylogenomic study to investigate the systematic position of Miltotranes in Molytinae and the evolution of cycad pollination of Miltotranes. I would like to thank many people for various assistance during the collection visiting, field works, providing photographs, and collecting permits. I also want to thank the Society of Australian Systematic Biologists and the ABRS for the student travel grants. Thank you for your listening.